Hello, my name is Debohuti, as was said, and today I'm here to talk about an original thesis on cancer treatment that uses elements of immunotherapy and oncolytic biotherapy. Of course, this project would not have been possible without my amazing mentor, Jude Raj, so special thanks to him. But to begin, we must first clarify the reasoning behind my hypothesis, which is that cancer is still a prominent issue. If we look to the right, there's a figure underlying leading causes of death, and that we could see that cancer is at the top of it. So the fight is not over, but I want to do my part in helping in the battle against it. So in order to fully define my theory, we must look at the foundations of it, which is immunotherapy being one, and breaking it into its respective parts is that immuno pertaining to the immune system and therapy, treatment intended to relieve or heal. Immunotherapy is essentially boosting or utilizing your immune system to search and kill target cells. This can also slow the proliferation of cancer cells, and some examples of these are monoclonal antibodies, immune checkpoint inhibitors, and cancer vaccines. And the great thing about using your immune system is that it has memory. So every time you successfully fight off a pathogen, your body makes immune system memory cells. So if you encounter that pathogen again, you will launch a quicker and more effective attack against it. So we can use this mechanism against cancer cells. Because a great problem is that even after you successfully fight off cancer cells, the cancer can still return. We can use this to combat that. And this is all great. So why isn't it the most used treatment? It is because that is only effective in a small subset of patients, which I intend to combat in my thesis. And the next foundation is oncolytic biotherapy. Oncolytic biotherapy is just the modification of viruses to prime the immune system to attack tumor cells. And I intend to use its characteristics of fewer drug-related side effects, precise targeting, and long-term durability. And also, it helps you distinguish between cancerous and non-cancerous cells if a unique biomarker or a higher density biomarker is given. And now onto my hypothesis. I suggest using technology constructed in the study outlined in the paper, Engineered Cell Entry Links Receptor Biology with Technology, used called lentiviral-mediated cell entry by engineered receptor ligand interaction, otherwise known as ENTER. ENTER essentially allows for receptor specificity-based gene delivery. So using these concepts, I propose a two-step process. Step one is the receptor. We're going to use a uniquely or highly expressed receptor in cancer cells, and this receptor can be tuned to the patient. One example I give in my paper is EGFR-positive lung cancer, where EGFR is in higher quantity, which is indicative of the presence of cancer. And then the surface display proteins can target this. This can also be used with other receptors like HER2 and GPR161. The baseline is that it's customizable depending on the expressed biomarkers. Step two is the payload. I propose using strains of previously given vaccines. Using once exposed strains allows for two things. Your immune system is already primed to recognize it, and you already have the memory cells for it. Means that once you encounter that pathogen again, it is your more effective and quicker attack. And what this allows for is also that this can be customized on a patient-to-patient basis. So we will look at popular vaccines like tetanus/diphtheria, polio, or MMR, and the viable contender will be based on after reviewing the patient's vaccination records and also their antibody titers for each virus. And to ensure an immune response occurs, the patient's T cells will be tested in vitro before administering the engineered virus in vivo. Unfortunately, there can be some drawbacks, which is that there can be difficulty in discerning a healthy cell from a cancerous cell, as I said. But the thing is, the expressed biomarker will be in higher quantity, so the virus is more likely to infect the cancerous cell. But if autoimmune symptoms do arise, we can turn to immunosuppressive drugs to mitigate the side effects and also suppress the immune response. And I intend to use these benefit of durability that the immune system gives, and also the universality that using any receptor gives in my thesis. Now I will open up the floor to questions.
Perfect. Thank you so much, Debohuti. That was really fascinating. We have another question. This is again, specific, someone was really listening. Can you elaborate on how, sorry, this is a long one for me. Could you elaborate on how you envision the integration of oncolytic viral therapy with current immunotherapy regimens in the clinical setting, particularly in terms of patient selection and treatment optimization?
So, oncolytic virotherapy is a modification of viruses. So, as I said, we're taking technology that where we modify a virus. So, on the top, on the outside is going to be the receptor based on the cancer, and on the inside is the payload, as I expressed, which is going to be previously given vaccines.
Okay, so it interacts with previously given vaccines. So that's the
Yes.
current immunotherapy regimens.
Yes.
Okay, great. Someone asks, can you elaborate more on what drew you to this topic of research?
So, I am actually in an accelerated program where I get to interact with different spots of medicine. So in my anatomy class, I was really drawn to this unit, especially because I want to pursue a career in medicine, and just seeing the fact that there's this big problem that has been here for a really long time, and we've been making many forms of treatment, but there hasn't been really a base treatment that works for everyone. So I wanted to make a form of treatment that could work for more people.
Okay, yeah. And this, we have another question from the audience. This overlaps with what, of course, I might ask you about continuing your research, but someone says, Debohuti, is this something that you see yourself continuing to research and learn about? You speak so well about the topic. I wonder if you have plans to keep pursuing it.
Definitely. In my college, I want to get access to better laboratories. Since I don't have access to that as a high school student, I could definitely test out some of the things I've said, because clinical trials are definitely required to see unforeseen consequences further and also see how this theory fares physically.
Okay, great. Thank you so much, Debohuti. Great presentation, really well spoken. Really appreciate the work that you've accomplished here.
Thank you.